Hello and welcome to this video on the first attempt at a completely synthesized eukaryotic genome. For the purposes of this research, researchers are using one of our favorite organisms, Saccharomyces cerevisiae, which is commonly known as yeast. Like the more advanced human body, the yeast cell has a great deal of overlap in the way it works. This ranges from the genes of similar size and function through to the way it modifies the products of these genes. Like a human, it has multiple chromosomes, but unlike a human, there are only 16. Synthetic genes are finding an increasing place in research. Where we originally used a comparatively simple process to stop a gene's expression, we are now looking at replacing whole genes. That was at first done by taking a known gene from one species and transposing it into another. The problem there is a simple but important limitation: finding a gene or genes that fit into the new organism. Initially, genes of little substance but great use were found and applied. The common example is the green fluorescent protein gene from jellyfish. This was able to be transferred to other organisms and provide a good indication of a successful gene transfer in a plasmid. It has later been used to create a commercial pet fish variety that can glow in the dark. This was useful, but it has the simple limitation of a long discovery time, and later the right applications. If we want to do something more or else, then we need to find a way to make it work. This must also be compatible with the organism in question. It's important to bear in mind that not all genes are the same, even if they perform the same function. Even if they perform the same function and are the same gene, they may not use the same set of DNA sequences. There is only a minor difference between the human, pig, and even mouse insulin, but it's enough that can create problems, with only the most minor of variations between species. This has led to the sequencing of genes from other organisms and figuring out why they are incompatible for the intended organism. These could be those small differences in the genome that mean the product expressed, if it is a protein, cannot work. This could be a slight change in the amino acids that make up the chain. This can be a long and difficult process. Like the first genes that were transferred, you can begin with a theoretically similar gene, then identify the differences. This can be done because every amino acid is coded for by a consistent codon or set of 3 DNA nucleotides in a sequence that always makes the same amino acids. If you know what goes in the sequence and what it creates, you can substitute for new codons. This makes a new product that could be compatible for the new intended purpose, but not the host making it. We can, at least in theory, transpose any single gene into a new host, but if it's incompatible and overrides an existing essential function, then we have doomed that new lifeform to death immediately. This whole approach has limitations anyway, and the simplest approach is down to errors. The problem with errors means that most genes, if we're going to do it using a simple insertion method, are limited to a shorter 200 nucleotide sequence. It's a very convenient size if you only want to swap out parts or whole, small, short genes. We use it regularly for silencing of genes by putting a nonsense mutation into them. This does create a problem though. A problem that can be overcome by taking the relevant parts of a gene, promoter, coding region, or termination sequence. If you can keep the promoter sequence, rebuild either it, the coding sequence, or the ending sequence, residue by residue, and then modify it, you can overcome this problem to some extent. We say some extent as you may only want to focus on one region at a time. That means you can insert small modifications somewhere in there that matches exactly. If you need to get bigger than this, then you need to look at other solutions. This can be done by creating longer sections of DNA that can be assembled together. This is called annealing. Think of it like Lego blocks. You can either build them one at a time or place a larger piece down. If you can't do either of those, try and find an intermediary. In theory, this works by building with a subunit of 50 nucleotide bases. This then gets shepherded into place by a chaperone molecule. This leads to much larger units that can get between 600 and 1200 base pairs in length. Some can be longer, but we still do not get anywhere near the yeast genome. This still leaves us a long way from the 12 to 14 million base pairs of the whole yeast genome, or the relatively small size of a single chromosome at 800,000 base pairs. This leads to a creation of a whole synthetic genome, or at least attempts at it via the Syntheic Yeast project. Yeast itself is an ideal organism for a few reasons. It has a well-established and fully sequenced genome. The organism's behavior and life cycles are well known. Yeast has been a model organism for decades, meaning resources, samples, equipment, and more are readily available, staff are well trained in them, and the model itself is often extremely robust, or at least the methodologies are. By contrast, smaller organisms with a less substantial genome are not as well prepared for this kind of study. This leads to the first steps of the Syntheic Yeast project, a chromosome. This is the second big step for the Syntheic Yeast project by itself. It is arguably one of the big first achievements here that we want to touch on, as it is the building block of these chromosomes that will lead to a whole organism later on. Remember, a chromosome is 800,000 base pairs in this case. This is still far too many for the earlier mentioned methods. What they did was to scale the idea to a ridiculous degree. They are using a similar idea, just executed very differently. They are using a building block of 750 base pairs. This then makes a 3000 base pair unit, which then in turn makes a 10,000 base pair unit. These then become 30 to 50,000 base pair units. These are inserted into the yeast in units of 3 to 6. They have a special end on them that makes this possible. To quote one of the resources they have, the termini of each 10,000 base pair chunk encoded restriction enzyme sites that enable directional assembly of chunks into so-called megachunks by in vitro ligation. This means they've created small fractions of the array that they have coded and made synthetically, so that it lines up with another end, giving a directionality. Think of it as like magnets. A negative will attract a positive, and the two will line up. Well, their synthetic genome has a similar feature with their 10,000 base pair units. They line up, and because they contain the same code, they effectively overlap. These sections and the resulting chromosome sharing significant overlap with the original sequence is important. It's important to keep the genome from mutating too much during meiotic recombination. Think of it this way: if you already have a good system, why break it? And that's what they've done here. Another way to think of it is if you have two recipes for a cake. Both rely on flour, milk, eggs, sugar, and other common items. There are minor differences beyond this, and this lets you swap out ingredients. But since you're not really swapping them out, given they are the same, it allows you to keep the same recipe and modify it. To further look at what they published on this, they were able to specifically use certain ligands that would break at specifically targeted sites. This breaking was important for selecting yeast that had the synthetic genes in there. The way they would do this was to create a lethal change in the genome if those breaks were not occurring at the right place, allowing it to come in. If they had their wild type sequences present, then those breaks were not there, and it would result in effectively a lethal mutation. This meant that there was selective pressure to ensure only the synthetic and semi-synthetic genomes were recombined to create the next generation of yeast cells. Not only that, but it ensured homology between them. Having not only created a synthetic gene and have it successfully put into the yeast, what they had to then make sure of was when it was copied and made a second copy of for reproduction, that they were getting the right things there. Now, they had a few options here, but again, why fix what's not broken? And in this case, they were able to use some of those markers they had installed for the proper joining of these genes and use it to ensure that there was specific checks and balances done by the cell's normal cell replication mechanisms. This would ensure that everything was done as it should be in the right direction with the right genes. Further to that, it allowed for a nifty little trick. What it did was that it took any regions that were thought to contain irregularities, create a small hairpin loop, which could then be cut off at the bottom. The DNA that was then let out would be degraded by enzymes, and the missing section would be replaced by the appropriate DNA checks and balances. All of this is great in a theoretical setting, but it has no use if it can't be translated to a real-world application. This meant that they've been testing their yeast to see just what happens. The common types of tests you could see are things that we've explained before: the growth, expression of various phenotypes, and morphology. Does it look right? Does it behave right? And ultimately, can it keep going? The way they did this was to use a variety of cultivation methods and a variety of stresses. This not only ensured it would respond to stimuli, but that it would predictably respond to stimuli, especially when compared to a wild-type sample. Wild-type is essentially their control group with no modifications made. What they found was that it was largely comparable. There were, however, a number of interesting phenomena they noted which led to it having some deleterious changes. This owes to how they've gone about assembling it. Remember that they make it by assembling various components, inserting them into the genome individually, having them reassemble spontaneously within there using the cell's methodologies and mechanisms, and then hoping that it all works. Naturally, there are some methods behind this that are meant to improve those odds, but this is also one of the reasons why one of their megachunks did not work the way they had expected. It had a problem, specifically that some of the places where it wound up interfered with other genes. Particularly, it was a methyltransferase gene. This has an effect on the yeast's ability to grow and how sensitive it is to heat. This meant that it did not perform as well in some instances. They particularly noted that at 37 degrees centigrade, in combination with glycerol, the colony was much smaller. They were able to track this back to those mutations associated with their synthesized genome. Now, naturally, it is still the early stages of this research, and we are nowhere near getting done. But there is far more to it than we could possibly describe in this video. The reason why we halt here is the processes beyond this point are either specialized, very specific, of limited interest to the broader audience, or deserve their own specialized attention in the future. Understanding what we've described so far is important for what will go on in the future. So far, they've already been able to get a synthesized genome, assemble it inside the yeast, and have the yeast perform reasonably well by comparison to a wildly occurring, completely natural yeast. What this means is something of interest and importance to you if you have gone this far. Think of what can be achieved by the stepwise modification of an entire organism's genome, being able to rebuild it from scratch and change it as needs be for an intended purpose. This may well sound rather monstrous, but it does really have practical applications. Think on the possible production value of homologous and bio-ready insulin, the reasonably recent morphine produced from genetically modified yeast. There are also cells that could be used to produce blood, skin grafts, and with some degree of hope, a universal donor type. Many things that, if we were to take this idea and extend it out, really do have significant applications. This week alone, we've already mentioned that there is a significant shortage of injectable opioids in hospitals, and much of this owes to the difficulty in transporting and supplying it for little profit. It's well established that there's a general shortage of organs, and the same thing goes with blood. Think of DNA as an instruction book. This approach lets you create the smallest manual with the most information efficacy. Think of a lean design that can be used to greatest effect. It involves removing unneeded genes, making certain genes more suitable for the purposes, or even adding in new features. For our own interests, this could be creating a yeast genome that is far more suited to creating ethanol-based fuel. As it stands now, it's reasonably efficient, but it has problems. In particular, getting an adequate yield in the terms of ethanol that can then be removed satisfactorily in an efficient manner. Starting with one single-celled eukaryotic organism, we can keep building up to something more substantial than what we see now, but this achievement by itself should not be underestimated. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.